Hi folks. Today's topic is Mendelian genetics, and of course genetics, to the extent that I understand it, which is very little, is an extremely complicated subject. We're going to just talk about the very simplest aspects of it that Gregor Mendel discovered in the 19th century. And it'll give you some idea of how you could generalize this to more complicated situations. But it's just supposed to be kind of a toy, almost a toy example of mathematical modeling. Okay, let me refresh your memories. I'm sure you learned this stuff in high school. But Mendel grew pea plants, and among the many characteristics of pea plants is the height. And he noticed that some of them were short and some of them were tall. And he noticed some things about them. For example, he noticed that if you had a short pea plant and you forced it to mate with itself, that is, the pollen taken from itself was put on used to form the seeds, then it would only produce more short plants. Whereas if you had tall plants, they could often, well, sometimes at least, tall plants could sometimes produce short plants as offspring, even when mating with themselves. So he hypothesized that there was some kind of signal in the plant that determined whether the plant should be tall or short, but he hypothesized because of this complication that short plants only produce short plants, but tall plants sometimes produce short plants. Right? Tall plants did not only produce short plants. He hypothesized that each plant actually contained two signals, and that if one of the signals happened to be for a tall plant, then that one was dominant and would express itself and the plant would be tall, whereas only if both signals were short would the plant be short. Okay? So he hypothesized that that these two signals, one S and one T, and I've written T capitalized because that's the one that's dominant, right? So if T and S are both present in the same plant, then it's the dominant one, T, that expresses itself and the plant will be tall. Now, in modern terminology, and please correct me if you're a real biology student, you actually know something about genetics, please correct me, email me, or in the office hours, please let me know if I'm making any terminological mistakes here. But in modern terminology, I think we say that in a situation like this, that the height of the plant is governed by a particular gene, and that gene comes in two alleles. These are variants of the gene, one for short and one for tall. And that each plant actually has a pair of alleles. It gets one from each parent. Plants also reproduce sexually. Plants have a stamen and a pistil. The stamen produces pollen, which finds its way to the pistil, and then the pistil contains the eggs, and the fertilized eggs becomes the seeds for the next generation of plants. But plants are actually bisexual. I'll mention that in a minute. So each plant, or at least with pea plants, I think there are some plants that come in male and female varieties, but I believe most plants are actually bisexual, so they have both stamens and pistils. And that means it's possible for a plant to reproduce all on its own, just mating with itself. So because a plant gets one allele from a stamen and one from a pistil, it actually has two parents. It gets one allele from both. So a plant has two alleles, and there are three possible combinations. I mean, assuming that only these two alleles are possible, S and T, then there are only three possible combinations, right? SS, TS, and TT. The order wouldn't matter, so TS would be the same thing as ST, right? Order is unimportant here. I don't know if it might actually be more complicated. I imagine that there might be other alleles, but maybe the other alleles are in some sense indistinguishable from S and T, so we don't really need to worry about them. Or maybe the whole thing about height is really more complicated than Mendel realized, which is probably likely. But anyway, we're going to stick with this simple model, because after all, it is just a model. We don't have to try to capture all of reality in perfect verisimilitude. Okay, so we're going to assume that there's only the two alleles, and that means there's only three what are called genotypes. So these are the three different combinations of alleles that a plant might have. Those are called the genotypes. Right? Now, because T is the dominant allele, any genotype that contains a T will result in a tall plant, whereas in order to get a short plant, you have to have both alleles be S. Right? So only S produces a short plant. The other two contain a T allele, which is dominant, so those plants will be tall. And warning again, genetics is usually very complicated. I know almost nothing about genetics, so feel free to clarify. Feel free to correct me if I make any mistakes, if you're a bio student. So as I said, a plant can mate with itself under selfing. So for example, what Mendel would do, I think, is he'd put a bag over a plant to prevent the pollen from getting out and reaching another plant, and also prevent any other plant's pollen from getting in. And in this way, he could ensure that the plant would mate with itself. And this is how he discovered that short plants that mate with themselves only produce other short plants, right? Because if a plant is short, it must have two S alleles, and if it has two S alleles and no T alleles, it can only produce more SS genotypes in the next generation. Whereas tall plants will sometimes produce short plants, because if a tall plant happens to be a hybrid, if it has both alleles, TS, then it can actually produce an SS offspring. Right? Now, you probably drew these diagrams in high school. So this is where you show what the parent plants look like, and then what the possible cases for the offspring are. So, for example, if the allele that comes from the stamen, well, if the plant from which the stamen comes is a TS plant, and if the plant from which the pistil comes is or is attached is a TS plant, then that means the stamen can provide either a T or an S allele, and the pistil, or the eggs in the pistil can also contain either a T or an S allele, and that means the offspring can be any of these four types, right? TT, TS, well, actually really three types, TT, TS, and SS, right? Again, we don't distinguish between TS and ST. So a hybrid plant, TS, that mates with itself can produce all three genotypes in the next generation, right? However, those three different genotypes only have only express themselves as two different phenotypes. I hope I'm using this lingo correctly, but a phenotype, I believe, is what you actually see expressed in the plant itself, right? How it actually expresses itself in the next generation physically, whereas the genotype is the genetic content of the plant, and the phenotype is what's expressed. So in the next generation, in this case, there are only two possible phenotypes, tall and short. Now, quantitatively, because the stamen has a 50-50 chance of providing either a T or an S, and similarly the pistil also has a 50-50 chance of providing either a T or an S. That means each one of these boxes here has a 1/4 chance of occurring, and therefore we can say that the overall probability of a TT plant in the next generation is 1/4. Same for an SS plant, because each one of those has only one box, whereas the TS plant, on the other hand, has two boxes, right? There are two boxes for the TS plant, so you add up 1/4 plus 1/4, and you get a 1/2 probability of a TS plant being produced in the next generation. Okay, so that's what happens when a hybrid plant, a TS plant, is selfed. That's the verb. We self it, or it performs selfing, so it can only mate with itself. And then the hybrid plant can produce three different genotypes in the next generation with these probabilities. Notice they're not all equally likely, right? It's most likely to produce another mixed plant, TS. Now, if you don't force the plants to self, then there are 6 possible things that can happen. So over here we got these 6 possible combinations that can happen. These are the 6 possible mating combinations, right? So any one of the 3 can mate with any one of the 3. Again, the order doesn't matter. It doesn't matter which plant is the stamen and which plant is the pistil. The offspring will be the same as far as height goes anyway, regardless. And so you can perform, you can draw a similar diagram, and again, I'm sure you did this in high school, you can perform a similar diagram with any one of these combinations. So for example, suppose the stamen plant is SS and the pistil plant is TS, and then once again you get these 4 combinations in the offspring in here. But in this case, this is rather simple. There's only 2 different genotypes that can be produced. You can either get a TS or an SS, and that's it. In fact, the hybrid case up here, this was the most complicated of all the mating combinations, right? All the other combinations are somewhat simpler. So down here in this case, we find that there's a 1/2 chance of getting a tall plant because all the TS plants will be tall, and we got 2 cases where the offspring are tall. Each case is probably 1/4, so that's a 1/2 chance of getting a tall plant, and similarly a 1/2 chance of getting a short plant, which only happens in the SS case. Right? Now, I should point out that here we're assuming equally likely outcomes, and I don't know, maybe that's not justified. Maybe for some reason, the germ seeds that have an S and a T, maybe they're not equally likely to propagate, right? It seems unlikely to me, but it's possible. It's something that you might want to investigate if you're a biologist. But again, this is where the model is not even trying to take that into consideration. In the model, we're assuming that every type of germ, stamen, a piece of pollen, or an egg, that they're all equally likely to fertilize or be fertilized. Right? Now, what we want to think about is what happens in the next generation. Suppose you have a population of plants, and suppose that they're going to mate at random, then what can we expect in the next generation? Well, let's see. Let's suppose, let me just set up the notation first. Let's suppose that in the current generation that a fraction P0 of the plants are of type SS, P1 fraction are of type TS, and P2 are of type TT. So these P's are basically the probabilities that if you pick a plant at random, it'll have the given genotype, right? So the P's have to add up to 1, right? They're like probabilities. And what we'd like to know is what kind of distribution of the genotypes could we expect in the next generation, right? If we know how the current generation is distributed according to genotype, then we should be able to predict what will happen in the next generation under the assumption that mating is completely random. Right? So what I do here is, now this is rather tedious, I admit. I go through all 6 of the mating combinations, right? These are the mating combinations. Calculate the probability that mating combination actually occurs. Right? Let me do that first. So the probability that 2 SS plants come together to mate and form a fertilized seed, right, would be P0 squared, right? Because each SS plant has probability P0 of occurring, and therefore the probability that 2 of them occur is P0 squared. The probability of an SS and a TS is 2 times P0 plus P1. Here we do have to take into account the idea that SS mating with TS is different from TS mating with SS, because both combinations are possible. Now we're talking about physical processes, so the stamen could be SS and the pistil could be TS, or the other way around, and we have to take both of those into account. Whereas as far as the genotype goes, it doesn't matter what the provenance of of the two alleles was, whether the S came from the stamen or the pistil, doesn't matter as far as the genotype goes. Okay, and then similarly SS and TT down here, that would have probability 2 times P0, P2, right? Because you could have an SS and a TT, or a TT and an SS. You could also have, let's see, 2 TSs, that would be P1 squared. You could have a TS and a TT, that would be 2P1, P2, and 2 TTs would be P2 squared. And by the way, you should find that if you add up all 6 of these probabilities that we just figured out, that they add up to 1, right? Because this is a list of mutually exclusive but exhaustive cases, so the probability should add up to 1, right? And they do. Now, within each one of these 6 cases, we have to further subdivide them according to what the offspring could be. Now, in the case of SS, the only offspring we get are more SSs. And that's also true in the case of the TT. So what we can say is that we expect this fraction, P0 squared, of the offspring to be SS. And similarly, we expect this fraction, P2 squared of the offspring, to be TT. Although, as we'll see, there are other sources of SS and TT, but there's at least this many. Now, let's see. What happens when you mate an SS with a TS? Well, actually, we looked at that over here, right? On the previous page, we looked at that case if we made an SS with a TS. And in that case, what you find is half the offspring are TS and half are SS. So I fill that in over here. Half are SS and half are TS. So what I did was I just took the probability of this mating combination is 2P0P1, and I multiplied that by 1/2 to get the probability of each one of these offspring. So this is going to, so that means this proportion of the offspring will be SS, this proportion will be TS, although again, as we saw, there'll be more in the future. There'll be more ways we can get those genotypes. Now when an SS mates with a TS, there's only one possibility. All the offspring are going to be TS. So I say SS and TT, all the offspring are TS. So all the 2P0 times P2, all of them will funnel into the TS genotype, right, in the next generation. The most complicated one was when you do a TS with a TS, and in that case, as we saw, there are 3 possibilities, and we saw that there's a 1/4 chance of SS, a 1/4 chance of TT, and a 1/2 chance of TS. So I just multiplied 1/4, 1/2, and 1/4 times the total P1 squared to get these probabilities over here. So those are going to contribute to the probabilities of getting SS, TS, and TT. And let's see, if we have TS and TT, well, that's very similar to TS and SS. Half go to TS and half go to TT, so that's how I got these numbers. I just took the 2P1, P2, and multiplied each one by 1/2 to get the probabilities of TS and TT. Okay, so what do we got? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so we got 10 terms in the different branches of the offspring. So let's add everything up. Total number of ways we can get SS genes. Let's see, maybe I should pick another column. How about this? Total number of ways we can get SS genes. Well, they came from P squared, that was this. Right? Also from P0, P1, that was this. Right? And from 1/4 P1 squared, 1/4 P1 squared was this, right? And so we add those 3 up, and what do you know? It actually factors. So P0 squared plus P0 P1 plus 1/4 P1 squared. You could actually rewrite that as P0 plus 1/2 P1 quantity squared. Okay, kind of cute. And now let me choose a different color. How about TS, right? How do we get TS? Well, let's see. One way is here. We can get P0 P1. Oh, all of these are TS, so we can get that. And then we can also get TS here. That was 1/2 P1 squared, and we can also get them here. So P1, P2. And when you add up those four terms, once again, gee, it seems like a remarkable coincidence, but when you add those up, it turns out that also factors. It actually factors into P0 plus 1 half P1 plus 1 half P1 plus P2 multiplied together and then times 2. Okay, whatever. Just noting that. And then finally, let me see, I'm running out of colors here. How about maybe gray? Okay. How do we get TT? Well, TT, there's three ways to get TT. Let me see, where's the first one? Oh, here's one. We can get TT this way. That was 1 half P1 squared. We can also get TT here. That's P1, P2. And we can also get TT here. That's P2 squared. And similarly, when you add those three terms up, that also factors into 1 half P1 plus P2 squared. Okay? So anyway, we did it. It was tedious. Yeah, it was so tedious that it might be worth looking for another method for doing this, huh? What do you think? So let's consider that on the next page. Is there an easier way to get the same result? And in particular, gee, we notice that these things factor, these expressions factor. Is there some way to explain why these expressions factor the way they do? Or to put the question differently, is there another way to derive these formulas for the probabilities for the next generation, the probability of each phenotype in the next generation? Is there a way to derive these probabilities so that we end up with these formulas directly, right? And maybe that would produce an easier way to do it. Well, the answer, of course, is yes. There is an easier way to do it. And if you're the type of person who likes puzzles, you might want to pause the video now and think for a moment about where these expressions might have come from. P0 plus 1 half P1. Notice that only two expressions occur here, right? P0 plus 1 half P1. And then P2 plus 1 half P1, right? So let's see here. Once again, I'm running out of colors. How about this? We've got P0 plus 1 half P1, and then we've also got 1 half P1 plus P2. Those are the two different expressions that we see here. So it seems that you can get all three of these formulas by using only those two expressions. How might you do that? Okay, so pause the video if you like puzzles and see if you can figure that out. Okay, so here's another way of thinking about exactly the same problem. So again, recall that P0, P1, P2, those are the proportions of each, or the fractions of each genotype, in the population, right? And they add up to 1. You can think of them as probabilities that add up to 1. Okay, so what we do in this case is we say, look, let's ignore the genotypes. Let's just think about the alleles. What proportion of the total allele population is S, and what proportion of the total allele population is T? Well, that's easy enough to figure out. Since the SS genotype is all S allele, right? So that contributes a fraction P0 to the S allele. Similarly, the fraction, the TT, is all T allele, and so that contributes a fraction P2 to the T allele. The TS genotype is split. Half the alleles are T and half are S, so I just split that up into 1 half P1 is S and 1 half P1 is T. So this is where those expressions came from. The P0 plus 1 half P1, that's the fraction of the alleles that are S, and P2 plus 1 half P1, that's the fraction of the alleles that are T. So we can say, let's look at simply the allele population, and then, what proportion are S and which proportion are T. Now, intuitively, this ought to be a perfectly valid way to compute the proportions in the next generation, because when you choose two genotypes at random and then mate them, right, which is the way we were thinking about it previously, you choose one of these, two of these three genotypes and mate them, Well, you're choosing the genotypes at random, and then you're choosing one of the alleles in that genotype at random also. So basically all you're doing is you're choosing a random allele from the population. And you end up with an S or a T allele according to these probabilities. So despite the fact that we're choosing a genotype first and then an allele, mathematically it's the same as just choosing a random allele from the population. So what we can do is we can compute the genotype composition in the next generation by just imagining that we pick one random allele, actually two random alleles from the allele population and combine them to form a new plant. So if you think about it that way, what's the probability that you end up with an SS genotype in the next generation? Well, that would happen if you happen to get two S alleles from the previous generation, and that happens with probability P0 plus 1/2 P1 squared. What's the probability of getting a TT genotype in the next generation? Well, that's the probability that you get two T alleles from the previous generation, and that's P2 plus 1/2 P1 squared. The probability that you get a TS genotype is, well, that means you can either get a T from one parent and an S from the other, or an S from the first parent and a T from the second. So in this case, again, we have to multiply by 2, but then we multiply the probability of getting an S and times the probability of getting a T. And so this does explain why we got those factored forms of the formulas on the previous slide, because that corresponds to just thinking about it in terms of alleles instead of thinking about it in terms of genotypes. Right? This method is much easier, right? So I advise you, if you see a problem like this on the homework or on an exam or something, use this method rather than the method on the previous page. I mean, I think the method on the previous page was useful in demonstrating a technique that might actually be necessary under different assumptions, but for the problems we're going to be asking, I think that this method is adequate and much easier. Okay, so to restate this, what we can say is that we can calculate the genotype frequencies in the next generation using only the allele frequencies in this one. In fact, the formulas depend only on the allele frequencies in this generation. Now, one consequence of that is that you don't really expect the allele frequencies to change over time. Right? You kind of expect the allele frequencies to remain constant over time, and that's because if you have if you start with a certain allele distribution, right, a certain fraction are S and a certain fraction are T, then you wouldn't expect that to change over time, because the next generation is formed just by sampling from the current generation at random. So you would expect the next generation to have exactly the same allele composition that the current population does. Now, there are a couple of reasons why that might not be the case in practice, in reality, in the real world. So this is a situation where our model is going to differ from reality, right? Our model is going to be a little too simple, simpler than the real world. In reality, maybe the most important thing to consider is that there might be selection pressure. Right? So if some characteristics of plants produce disadvantages in survival or in reproduction, then that's selection pressure, and then plants that have certain alleles may be less likely to reproduce than others. And in that case, we could expect the allele frequency to change over time. Right? So, to be precise, we should say that in the absence of selection pressure, if you imagine that there's no selection pressure, then we would expect the allele frequency to remain constant over time. And of course, this can happen, like, for example, in a greenhouse, right? If you're in a greenhouse, basically the plants are all being tended to by what do you call a guy who raises plants? A florist. And Therefore, there would be no selection pressure. Even plants that have a hard time surviving in the real world will be able to survive in a greenhouse, right? Because the florist wants them to survive. If you're cultivating plants in that way, then we could assume there is no selection pressure, and then in that case you would assume the allele frequency would remain constant over time. One other thing you might want to consider that might cause the allele frequencies to vary over time would be just random processes do not always produce the expected value, right? You might the average number of genotypes of each type, and the average number of alleles of each type in the next generation should be the same as they were in the previous generation, but the average won't always be attained because in a random process things don't always work out exactly, to the average, right? You can flip a coin 10 times, you don't always get 5 heads and 5 tails. So they're just random effects that might cause that to vary. And again, in this model, we're oversimplifying in that respect, and we're going to ignore that effect. Okay, so nevertheless, with all these assumptions that represent oversimplifications of reality, we now have arrived at something which is kind of interesting. It's called the Hardy-Weinberg Principle. It says that in the absence of selection pressure, as we've already seen, in the absence of selection pressure, the allele frequencies will remain constant. But because the allele frequencies remain constant, and because the genotype frequencies in the next generation depend only on the allele frequencies, that means the genotype frequencies also do not change over time. So in the absence of selection pressure, we would expect that the distribution of genotypes would remain constant over time. Okay, so that's an interesting fact. Now, one exception might be in the very first generation, you might start off with an artificial combination of genotypes. So the genotypes in the second generation may not be the same as in the first generation. However, once the alleles have had a chance to mix that first time and produce the second generation, from that point on, you should find that the genotype population remains constant, that the distribution of the genotypes is fixed over time. Right? Again, in the absence of selection pressure. Okay. So let me just point out a few things where the model differs from reality, right? So it's always a good thing to keep in mind how we're oversimplifying things. In the model, our model is going to obey the Hardy-Weinberg principle that the genotype distribution will be constant. That will not be true in reality. In reality, the genotypes will change in response to selection pressure and also to, as I said, to just random effects. This is sometimes called genetic drift, I believe, in the context of genetics. So we say that genetic drift is when there's just a random alteration in genotype distribution over time that's not due to selection pressure. It can happen just due to randomness. Okay. Another thing, one of the things we're assuming here, in order to arrive at the Hardy-Weinberg principle, was we were assuming that mating was occurring completely at random, and that mating occurred, that all outcomes are equally likely under mating, that any two genotypes are equally likely to mate, and given a mating combination, any two alleles are equally likely to combine in the offspring. And that also may not be true, right? So there may be selection pressure, there may be pure, there may be reproductive advantages in certain genotypes over others. There may just be things like propinquity, like how close two plants happen to be to each other may influence whether they're likely to mate, right? So simple things like that. There might be, wind direction or something. Maybe the wind happens to flow in one direction rather than another, and that might influence which pollen gets distributed to which pistils, right? So all of those things mean that in reality, mating will not be at random, and the genotype distribution will not remain constant. Nevertheless, the model can still be useful. Well, it can be useful certainly in cases where there is no selection pressure, right? As we talked about, like a greenhouse, right, where you're raising plants. But also, you can use the model to test how salient selection pressure really is, right? You can make a prediction based on the model, which assumes there's no selection pressure, and then when you see differences in reality from from what's predicted by the model, then you can conclude, you may conclude that you may be able to conclude that those differences are due to selection pressure. So this could be some evidence of selection pressure on a genotype distribution, for example. Okay, one last thing we should talk about. This is actually a simplification of what we just did. We just looked at the case when mating occurs at random, but I pointed out that a very useful thing to do if you're cultivating plants or cultivating farm animals or anything like that is selfing, is restricting the mating possibilities, right? So in particular with plants, what you can do is you can just put a bag over each plant to make sure that it mates with itself, called selfing. And that simplifies the problem quite a bit. If a plant is only mating from itself, then let's see, there are basically three things that can happen, right? If you start with an SS plant, it can only produce SS offspring. If you start with a TS plant, then it produces 1/4 SS offspring, 1/4 TT offspring, and 1/2 TS offspring. If you start with a TT plant, it only produces TT offspring. So, suppose we start with a population, using the same notation as before. Suppose we start with a population which has a fraction P0 of SS genotype, P1 TS, and P2 TT. Then all the SS plants are going to produce SS offspring, right? So all of these plants up here are going to produce, all of these plants will produce SS offspring, right? All of the TT plants will produce TT offspring, right? So they all contribute there. However, the TS plants, as we saw, these produce all three different kinds. So they split up into the three different categories, right? And therefore, in the next generation of selfing, you're going to find that the proportion of S plants is P0 plus 1/4 P1. The proportion of TT plants is P2. In other words, the same as it was before, plus it gets 1/4 of the hybrid plants from the previous generation, 1/4 P1. Whereas these hybrid plants, the TS plants, always gets cut in half, right? Of the TS plants, only half of those offspring will also be TS. So the pattern you see is that the TS plants, this fraction of the population is always cut in half, right? So the TS proportion is always cut in half under selfing. And of the other half of the TS plants, they get split up between the SS and the TT plants, right? So they're constantly growing, right? So the pure genotypes are constantly growing, whereas the mixed genotypes are constantly being cut in half. So what happens is, under selfing, you make every plant mate with itself, and you very rapidly approach a population in which all the plants are, or almost all the plants are basically pure. They're either all SS or they're all TT. And that's a good thing, if you're cultivating plants, that's a good thing to know that you have plants that are all SS. Well, actually, that's easy to tell. In this case, we know that any short plant is a pure SS. But it's not always clear when a plant is pure TT. So it's good to know that by selfing, you can be pretty confident that in the long run, all the tall plants are going to be pure TT plants. Right? And in fact, the longer the lineage of tall plants, the more confident you can be that the new generation of tall plants will in fact be pure TT. Right? So that's a very useful thing to do, to be able to produce a population of pure SS and pure TT. Okay.